All right. Well, hi, everyone. It's good to talk with you again. And thanks to Hunter's Hope for inviting me to speak. I'm going to give you an update on the cell therapies that we're developing to treat kids with brain injuries and leukodystrophies, and specifically talk about the cell therapy we call DUOC. I'll just mention that the work I'm describing is done in a center that we have at Duke called the Marcus Center for Cellular Cures, and we are a center focused on bringing things from bench to bedside, we specifically focus on therapies we develop here that we bring in to the clinic. And we have a portfolio of cell therapies that we are bringing forward and testing in a variety of different conditions, and they involve straight cord blood, particularly from a donor, where we're focusing on the monocyte as the active cell, or DUOC, which I'll talk about, which is a microglial-like cell we manufacture from cord blood monocytes, or cord tissue MSCs, which are third-party cells. And all of these cells have different capabilities in terms of treating leukodystrophies and other brain injuries, as shown in this chart. Many of them can reduce inflammation, which has applications in many diseases. They can promote oligodendrocyte proliferation, which then promotes remyelination. Some can modulate brain connectivity. And the diseases that we are studying are listed on the bottom of the slide, but include hypoxic brain injury, cerebral palsy, autism, stroke, leukodystrophies, multiple sclerosis, and even osteoarthritis of the knee. So today I'm focusing on cord blood, which, although you may hear people refer to as a bag of stem cells, is not a bag of stem cells. Cord blood does have the hematopoietic stem cells, which can rescue the bone marrow and the immune system after myeloablative therapy. But those cells really represent roughly 0.001% of the cells in the bag of cord blood. There are many other cells in cord blood which are now being developed into therapeutic products. I'm going to focus on the monocytes, but I'll just mention that the lymphoid compartment also is becoming sourced for development of allogeneic CAR T cells and CTLs and NK cells and other therapies. In our work focusing on the monocytes, we've been able to show that the fresh monocytes from cord blood, so not cultured or manipulated, can mediate brain repair in hypoxic injury, cerebral palsy, and autism, and that this cell DUOC that we manufacture from cord blood can induce remyelination. And I'll show you the work we've done in leukodystrophies, and we're planning trials in multiple sclerosis. So a lot of this is published, and I will share my slides later, but I just want to mention that we've looked into detail about the mechanism through which cord blood monocytes rescue brain cells after hypoxic injury in both in vitro and murine systems. And we've been able to show that this activity is not present in adult monocytes. It's really unique to cord blood. And it may be because some of the cord blood monocyte population is derived from yolk sac rather than bone marrow. And in the upper panel, you can see a brain slice model using murine brain that we've developed, used in our laboratory. And normal brain looks like the picture on the left, where the green cells are neurons, the red cells are oligodendrocytes, and the pink cells are astrocytes. The middle panel shows what happens if you shock the tissue with the oxygen glucose deprivation. 3 days later, you see a big scarring with the astrocytes proliferating and the neurons disappearing. But if you do the same stress, but you also add back cord blood monocytes, then you rescue the tissue on the right. And you don't have loss of neuronal cells or neuronal death. We took this observation into multiple clinical trials, including a clinical trial in children with cerebral palsy who had cerebral palsy because of an in utero stroke, prematurity, or birth asphyxia. And in these kids, we were able to show that they improved function, and that was associated with the dose of cord blood cells we gave. And in children with improved function on the bottom, you can see composite MRIs showing the motor tracks on fiber tracking. And if you see a red line or a turquoise node or circle, that means that there was statistically significant increase in the node size or the tract integrity or thickness, and that we hypothesized that the monocytes signal through cells actually in the body called macrophages that eat them, and then that sends signals to the brain to increase connectivity. And just to show you what that looks like in a patient, this little boy coming on study is spastic, walking with a walker, scissoring, cannot walk independently, and this little boy a year later is able to walk independently and no braces, much better balance. And that kind of change, although this is a child with cerebral palsy, I think is also going to be possible in children with damage to their motor tracts, for example, because of leukodystrophy, if we can harness the power of this therapy. We showed many years ago that unrelated donor cord blood transplants for children with inherited metabolic diseases and leukodystrophies could be an effective therapy, particularly if they were transplanted early in the course of their disease. You hear this theme over and over again, whether we're talking about gene therapy or enzyme therapy or chaperone therapy or transplant or gene therapy. The red bar shows you survival at 12 years of children who were transplanted early in the course of their disease, compared to the green line, which shows you the survival of children who came to transplant with symptoms. And because of this observation, which was made many years ago, in general, children with advanced symptoms are not considered candidates for transplantation. Just to show you some ways transplant can be effective. This is a youngster with Hurler syndrome. Those of you may or may not know that with severe phenotype Hurler syndrome, children generally die at 5 to 6 years of age. They die of cardiac disease with severe motor and intellectual impairment. And we showed many years ago that if children with Hurler were transplanted in the first 2 years of life, in the dotted black lines, that their IQ would improve and would get back into the normal range by the time that they were ready to go to school. We now know that the earlier a child with Hurler is transplanted, the better the IQ and other effects are. These effects are sustained. So I always like to say that I'm old enough to have followed patients for a long time now, and both of the girls in these pictures have Hurler syndrome. They're going to their senior proms. They were transplanted in the first year or two of life, and are now both in college. So this cognitive and other effects are sustained. For Krabbe disease, Maria Escolar and I and our teams published years ago papers showing that for pre-symptomatic newborns, survival could be excellent after transplant, and compared to symptomatic or untreated patients, both a survival benefit and a functional benefit could be seen in the disease. It's important to point out that these newborns were all identified through a family history of another affected child. Boys in the picture are two boys with late infantile Krabbe disease who were both transplanted early because of diagnosis in an older sibling who unfortunately died. Heather Alwelt and I published a few years ago a paper looking at functional outcomes of newborns transplanted with early infantile Krabbe disease. And we looked at function as a way of really knowing what these kids were like and also studying quality of life. And we were able to make a few observations. One, that most of these children survive and have a good quality of life, although many have disabilities, and that we saw less deficits in children who were transplanted before 30 days of age compared to those transplanted more than 30 days of age, although the more than 30 days of age was just a few weeks later. So these are not late transplants. And this is just a figure from that paper where red is abnormal, green is normal, and we're just looking at 3 domains: mobility, communication, and feeding. But the top bars show the children transplanted at less than 30 days of age. The bottom bars show the children transplanted at more than 30 days of age. And you can see the distinction and the fact that that 30-day window, which is somewhat arbitrary, but what we had to work with, is important, and that getting that graft in before 30 days will yield better results long term. Maria Escolar published a paper looking at 7-year and 15-year outcomes of children transplanted for early infantile Krabbe disease, and again showed that although there's great improvement in the cognitive and communication functions in these kids, most of the kids continue to have some degree of moderate to severe motor impairment. And I think we've all observed this in the majority of kids who've been transplanted at a young age, and that means there's still room for improvement. Just to illustrate that point even further, these are 2 girls, both with a family history, both identified in utero, both transplanted early. The little girl on the right had a transplant at 21 days of age. The little girl on the left had a transplant at 19 days of age. Just for those of you who think about genotype, the girl on the left is a homozygous 30 kb deletion patient whose sister died between 2 and 3 years of age. But Maria looked at fiber tracking of the corticospinal tracts of these kids, and the little girl on the left has a normal tract at 2 days of age, whereas the child on the right already had damage to her tract at 2 days of age, which again says that no matter how early we do transplant, we can't, unless it's in utero, we're not gonna definitely correct damage that occurred before birth. Now we in our work at Duke had looked at the brain of a child who had a transplant for symptomatic early infantile Krabbe at 10 months of age, who died at 20 months of age, so 10 months post-transplant, to really see if we could document engraftment of donor cells in our brain. And in this particular setup, the donor was male and the recipient was female. So this happens to be a stain of her cortex, which was done by Evan Snyder, who's now at the Burnham. But it's cortex with a DAPI stain, which is blue to stain the nuclei, and XY fish to stain the chromosomes. So anywhere that you see a turquoise or green dot, that's a cell with a Y chromosome, and that's a donor cell. So in this child, 10 months post-transplant, we did see significant engraftment of donor cells all throughout the brain, showing you cortex, but we had pictures from all regions. And the cell that we saw engrafting was predominantly a microglial cell. So we went back to the lab and said, can we grow that cell and make it into a cell therapy to augment transplant? We did, and the cell is called DUOC, and swimming around on the right-hand picture is this cell under time-lapse photography sped up. So it's a macrophage-like cell. It grows as an adherent cell. It takes on many different morphologies in its life cycle, but it's derived from the CD14 monocyte population of cord blood. And we now manufacture this cell and have an IND and have it in a clinical trial in children with leukodystrophies to bridge the gap of engraftment in the periphery and engraftment in the brain, which we know takes longer than engraftment in the blood. We studied this cell in many different ways, but its interesting features include the fact that it can remyelinate brain in demyelination models. So this is a model called cuprazone, where you feed mice cuprazone, which is a copper chelator, and that leads to demyelination of the corpus callosum. The mice are fed cuprazone for 5 weeks, then we stop the diet. The the corpus callosum is demyelinated, and we can do interventions and look at whether we can accelerate remyelination of the corpus callosum in the model. So this just shows you mice, an example of a mouse treated that way. In the control, they just got a sham injection of saline, and the brain remains demyelinated on the left and middle and upper panels. And on the right, you can see electron microscopy, which shows very distorted rings of mine. On the bottom, you see a brain from a mouse injected with DUOC, where 1 week later, the brain is remyelinated. On the left, you see Luxol fast blue. In the middle, you see no astrocytosis. And on the right, you see nice normal rings of myelin. We also have been able to show that the reason the brain is remyelinating is because the DUOC cells stimulate proliferation of resident oligodendrocytes from the recipient, and they actually are responsible for remyelinating. And in a clinical model, which is really used to study MS, called the EAE model, where we can induce an ascending paralysis, we're able to show in the blue lines that the DUOC cells given to early symptomatic mice prevent progression of the ascending paralysis. And that is associated with both decreased inflammation and also remyelination of the damaged spinal cord tissue. DUOC does many other things because it's a macrophage. It can clean up debris and pieces of dying cells that often cause secondary inflammation in the brain. It modulates and downregulates inflammation by secreting IL-10, IL-6, and TGF-beta. And again, I showed you that it promotes myelination. This is the molecular signature of the cell that we think is important, and we use that in manufacturing. And we went into a phase 1 study now 4 years ago, testing DUOC as an adjuvant therapy in kids with leukodystrophies undergoing cord blood transplantation. Initially, we used DUOC from the same donor cord blood unit, or we manufactured it from the same donor cord blood unit that we used for transplant. So this on the lower right is what a typical cord blood unit looks like frozen. And we use the big compartment for the IV administration of cells post-chemotherapy for rescue of the bone marrow and lymph and the immune system. And then we use the small portion for manufacturing of DUOC, which takes 3 weeks. Later, we advanced the trial to use 2 different cord blood units so we could get to a higher dose and more consistent manufacturing of DUOC. And for this trial, we capped the dose, which was given intrathecally at 10 million cells. The eligibility was standard for cord blood transplant for leukodystrophy, with the exception of the fact that we allowed children with more disease to be eligible. So for our standard transplant, the Lansky has to be 80 or above. And for this trial, the Lansky can be 40 or above. This just shows you the schema of taking the cord blood unit, giving the child chemotherapy, using the larger portion or a second unit for administration on day 0, and giving it to the baby. And then after engraftment, generally 28 days later, we give DUOC. DUOC takes 21 days to manufacture, so we start manufacturing a week out. And again, originally it was with the same unit, now it's with the second unit. And on day 28, we harvest it and prepare it for intrathecal administration and give it to the baby or the patient through a routine spinal tap. The manufacturing of DUOC occurs again in a GMP clean room setting, the cells grow in adherent fashion in tissue culture flasks, which are fed various types of media over the 21 days. And then on day 21, the cells are trypsinized, washed, and formulated in Ringer's lactate, which is safe to give into the spinal fluid. This just shows you the demographics of the patients we treated. So they range from 3 weeks to 15 years on the study. And although the median Lansky was 90, it did range from 40 to 100. And 74% were male, 81% were Caucasian. The distribution of diseases are shown here. We enrolled 34 patients Excuse me, and we've treated 28 to date, including one who was treated on an EIND 6 years post-transplant. A few children did not receive DUOC, even though they were enrolled in the study. 2 were critically ill at 28 days and died of TRM later on. 1 had graft failure, and 3 products had manufacturing issues when we were using the small fraction. The average manufacturing we got to was anywhere from 20,000 to 9.6 million cells with the single unit and with the small fraction. And with the single unit, we typically manufacture between 20 and 60 million cells, but we're capping the dose at 10 million cells for this study. Although we have a funded planned study in adults with MS, where we will take the dose up to 60 million cells. 25 of the 28 treated patients are surviving with a median follow-up of 28 months. There were 3 deaths. 1 patient with ALD died 3 months out of GVHD. 1 patient with advanced MLD, who was the sibling of a second baby we transplanted on the study, died from progressive MLD, and 1 Batten disease patient died in her sleep years out of unknown causes. So in summary, DUOC is a novel product. It's derived from the monocyte lineage of cord blood cells. We established manufacturing procedures for a first-in-man phase 1 clinical trial and treated 28 patients to date. We can utilize same donor or third-party donors to manufacture DUOC, and we continue to enroll patients using a second donor and have amended the protocol to administer DUOC years post-transplant. That amendment is held up at the FDA because of COVID, but we plan to execute that soon, hopefully in the fall when we're allowed to see certain types of research patients at Duke again. And we are planning and have been funded to do a separate standalone clinical trial in adult patients with MS to assess efficacy. We found it's really impossible to assess efficacy in the transplanted patients because we can't separate any contribution of DUOC that might be there from the actual transplant course. So in summary, in the bigger sense, transplant can rescue children with inherited metabolic diseases if they're transplanted early in the course of their disease. Early diagnosis and treatment yield superior results. And in the case of Krabbe, with the early infantile form, we're really talking about getting to transplant in the first month of life. While life's extended and quality of life is improved, full disease correction is not achieved. In Krabbe kids, peripheral nerve disease progresses, particularly in the second decade of life. DUOC is a novel microglial-like cell promoting myelination of the brain, which may improve disease outcomes. And I really think at the end of the day, combination therapies need to be explored to further improve outcomes of kids with these diseases. And want to thank all the patients and their families who participate in all these studies and work with us every day and work with all the investigators to try to make outcomes better for all of the kids with Krabbe disease. And again, it does take a village. We have a large team at Duke and many external collaborators who work on all these studies. Just want to call out Tony Filiano and his group in our research lab who have done all the preclinical studies on DUOC, and then our GMP team who do the manufacturing, our regulatory team who handle our INDs and all the interactions with the FDA, and then Jessica's son, Kristen Page and Manoj Prasad, who are my colleagues in these studies and helping to treat the patients. For the DUOC studies to date, we've had funding from the Marcus Foundation, which has funded all the patients treated on the trial to this point. So I'll stop there, and I